Good morning to everyone and welcome to this webinar. My name is Silvina Josifova and I work as a Scientific Program Manager in the European Pharmacopoeia Department. Today, together with my colleague Amela Sracevic, we will guide you through the different sections of an individual monograph. We will focus on monographs on chemically defined active substance and on medicinal products containing chemically defined APIs. The purpose of a monograph is to ensure patient safety. This means ensure an appropriate quality of the substance for pharmaceutical use. The monographs are elaborated for substances in products already assessed and approved by competent authorities in a member state. The European Pharmacopoeia monographs propose validated analytical procedures for the control of impurity profiles in different manufacturing routes for companies approved on the European market. It is essential to have an active collaboration with the manufacturers in order to have a good quality monograph. Yesterday, you have heard a lot about the general concepts, general notices, general chapters, or general monographs. It is important to remember that general and individual monographs are complementary. There is not an overruling, and if there are any exceptions, they are clearly indicated in the general or the individual monograph. On the screen, you can see the first part of the monograph of diclofenac sodium. It is a screenshot from the online version. On the upper left side, there is a link to the general notices, to the general monographs, but also to knowledge database. The knowledge database is a direct link with our users, where you can find useful information about regions or brand name of columns used during the experimental verification of the monographs. I would now propose you to start with a short quiz. May I ask you to respond to the question, which are the mandatory sections in a monograph? Please remember that several replies are possible. Please use Slido. You can scan a QR code and then respond to this question. I will wait a few minutes. Oops, sorry. So thank you for your active participation. I see that many of you are already very active this morning. This is much appreciated. So thank you very much for your participation. I see that many of you knows quite well the monographs, tests and essay mandatory sections. Let us see now the answers. All the sections that you see which are in green are mandatory. These are definition, production, identification, tests, and essay sections. However, there are informative sections, and those are given in orange: character storage and functionality-related characteristics. Please remember that in order to demonstrate compliance with the European Pharmacopoeia monograph, you have to satisfy to all mandatory parts of a monograph. The informative parts, informative sections, are not an analytical requirement. In the next 2 slides, I have summarized all the sections that an individual monograph may contain. This is a summary, and now we'll start to discover each and every section. Let us start with the title. The international non-proprietary names are almost universally used as titles of our monographs. They are modified to indicate the salt, and they also include the degree of hydration. For a well-defined hydrate, you will find the term hemi, mono, D3 hydrate, etc. in the title of the monograph. If a monograph covers a mixture of hydrates, then the terminology of hydrate would be given. In the gray part below, you will find some examples where you can study and ask questions after this presentation. The definition section contains the chemical nomenclature and the content limits. The content limits are defined by the analytical procedure described under essay. They are always expressed on anhydrous or dried basis, depending if a Carl Fisher test for water or loss on drying are described in the monograph. The solvent-free substance concept is always implied, even if it is not stated in each and every individual monograph. This concept is covered by the General Monograph 2034, Substances for Pharmaceutical Use, and by the General Chapter 5.4, Residual Solvents. For an HPLC assay, the limits would reflect the variability of the method and the purity of the substance. Thus, for a substance where a 2% of total of impurities is found, the lower limit can be decreased down to 96.0%. The upper limit would be set to 102.0% to reflect the variability of the method. The usual limits for a volumetric titration are 99.0 to 101.0%. And in some monographs, you can find a microbiological or a biological assay with respectively minimum or specific activity, which are described. I have just mentioned the usual limits for the volumetric titration. However, the limits will be set taking into account the type of the titration, but also the results from the repeatability and the relative accuracy during the experimental verification. The decision table that you see on the screen is part of the technical guide for the elaboration of monographs, which is freely available on our website. For an HPLC assay, the users have to apply reference standards with assigned content, but they have also to fulfill repeatability requirements, and those are described in the general chapter 246. We can again see the complementarity between individual monographs and general monographs or chapter. I would like to emphasize that this general chapter 246 has been recently revised and is now harmonized with USP and JP. Then I can encourage you to refer to the latest harmonized version. The definition section would also contain statements on the scope. This means information about the route of synthesis or about the degree of hydration. Every time a monograph covers a well-defined hydrate, a mixture of different hydrate forms, or a substance which is water-free or a hydrate form, you will find a different statements under the definition. I have mentioned every time different examples that you can study after this presentation. It is important to remember that the monograph applies to all grades, to substance for human and veterinary use, unless it is otherwise stated. Those must be of appropriate quality for the elaboration of all pharmaceutical forms, unless otherwise stated. And if there are any special requirements for special grades, such as the use of the substance for the parenteral preparations, then the information would be indicated in the individual monograph. The production section is not systematically included into an individual monograph, but when present, it is mandatory. It contains instructions for the manufacturers with respect to the source material, the manufacturing process, the control, validation, or in-process testing. This section cannot be necessarily verified by an independent analyst, and its compliance is established by competent authorities, either by inspection or through the examination of data. And this is the place when in some monographs you can find information about the control of DNA reactive impurities. Please remember that the absence of such a section in a monograph does not imply that the attention to above features is not required. Let us see together two examples. In case of ticagrelor monograph, a particular attention is paid to the formation of stereoisomeric impurities. In this case, the manufacturers have to implement suitable procedures for the control of those stereoisomeric impurities. In the case of pethidine hydrochloride monograph, particular attention is paid to the control of a certain impurity, code impurity B, at a maximum 0.1 ppm, and this when the substance is used in parenteral preparations. A few words about the character sections. This is only informative and is not an analytical requirement. However, this section contains useful information for the analysts with respect to the appearance of the substance, the solubility, or some physical properties, such as melting point or density. This is the section where you can find information for the existence of polymorphism, if known. Please remember that by default, an individual monograph covers all polymorphic forms, unless it is otherwise mentioned or stated in the monograph. There is a general chapter on polymorphism, 5.9, and also a general chapter, 5.11, where you can find methods for the determination of hygroscopicity, crystallinity, and solubility. Some information that might be of interest for you. The general chapter 5.11 has been recently revised and information has been transferred from general notices to this chapter. As of 11th edition, the terms ethanol and alcohol without qualification have been replaced in monographs by respectively anhydrous ethanol and ethanol 96%. We have characterized and defined our substance. We need now to identify it. The identification section is mandatory and it may contain two series of identifications, first and second. Those are defined in the general notices. The first identification series may be used in all circumstances. It is intended for pharmaceutical industry. The second identification series is intended for community and hospital pharmacies, and its implementation is subject to national regulations. Sometimes there might be cross-reference in this section to tests. You will also find a reference to water or loss on drink if a monograph covers a hydrate substance. Let us see more in detail the first identification series, intended for pharmaceutical industry. It contains tests such as infrared, HPLC or TLC. In some monographs, you may find the statement carry out either tests AP or test CD. Please do not mix this statement with the second identification. Those two sets of identifications given in the first series are equivalent and may be used independently. Let us see a concrete example. In case of phosphinopril sodium, 3 tests are described in the first identification: specific optical rotation, infrared spectrophotometry, and a chemical reaction of sodium. Those 3 tests have to be performed in order to comply with the requirements of the first identification series. In case of levetiracetam monograph, the user has a choice. The user can perform either specific optical rotation and infrared spectrophotometry, or choose to perform enantiomer purity and infrared spectrophotometry. The second series, intended for community and hospital pharmacies, contain different tests with much less complex instrumentation. Those can be TLC tests, chemical reactions, or mixed melting point. In the case of prednisolone acetate monograph, the second identification describes 2 tests, TLC and a chemical reaction, and those 2 tests have to be performed to comply with the requirements. The European Pharmacopoeia monographs propose chemical, physical, and chromatographic methods for the control of different impurities: organic, inorganic, volatile impurities, etc. Let us start with the organic impurities, which control is in line with ICH Q3A. There are 2 types of impurities: specified and unspecified, or other detectable. The specified impurities are detected, they are identified by suitable reference standards, and they have their own individual acceptance criteria. The unspecified impurities are known to be detected by the methods in the monograph, but they are not individually identified. They are limited by the limit for unspecified impurities given in the monograph. In the General Monograph 2034, you will find a table where the different reporting, identification, and qualification thresholds are indicated, depending on the maximum daily dose of the substance. Please remember that the relative retentions are only given for information. By default, they are not used to identify the impurities. These 2 types of impurities are described in an impurities section, or so-called transparency list. This list is non-exhaustive, and it contains the impurities which are known to be controlled by the tests described in the monograph. In most of the cases, the impurities are controlled by the related substances test, but sometimes other tests may be used for the control of certain impurities. This could be, for example, a UV absorbance ratio. Please remember that this section is established based on information obtained and experimentally verified during a monograph's elaboration or revision. You may ask yourself, what happens if you have an impurity which is not in this impurity section? Do you need to control it or to ignore it? And this case is covered by EU Directive 2001/83 as amended. If there are impurities which are not controlled in a pharmacopoeia, then the marketing authorization holder has to propose suitable procedures and tolerance limits to competent authorities. There are also cases that a specification described in a monograph might be not sufficient to ensure the quality of the substance. And in this case, marketing authorization holder would be requested by competent authorities for more appropriate specifications. In any case, competent authorities will inform the pharmacopoeia, and the marketing authorization holder shall provide to the authorities, but also to pharmacopoeias, with data and samples. This will also allow us to update our monographs. This part is included in the general notices. The limits of the impurities in a monograph are set taking into account the approved specifications and also batch and stability data provided by the manufacturers. In old-style monographs, you will find very often those limits expressed in area comparison style. While during revision of those monographs and during the elaboration of new monographs, we try to implement the quantitative expression. In area comparison expression, the users have to compare areas and report pass or fail results. In those cases, the numerical values given in brackets are only for information. On the contrary, in the quantitative expression, the users have to report impurities as numerical values, the levels of the impurities as numerical values, and have to take into account not only the areas, but also the concentration of solutions. And of course, in this case, limits are much more precise. Let us see together another group of impurities, the inorganic impurities. Those might be filter materials, inorganic salts, or elemental impurities. They result from the manufacturing process or from the raw materials. The biggest group of inorganic impurities are the elemental impurities, and their control is covered by ICH Q3D guideline, which is partly reproduced in the general chapter 5.20. In certain individual monographs, you can find some specific tests for the control of elemental impurities. Different tests such as inductively coupled plasma or X-ray fluorescence or atomic absorption spectrophotometry can be described. In case of cisplatin, for example, silver is controlled at maximum 250 ppm. In calcium acetate, magnesium is controlled at maximum 500 ppm. Almost all individual monographs contain a test for sulfated ash, and this test is intended for the determination of the total foreign cations present in the organic substance. I would like to pay a special attention to the elemental impurities. In general, you will not find a test for elemental impurities class 1, class 2A or 2B or class 3 in an individual monograph, unless it is necessary to ensure the quality of the substance. The tests for those elements have been suppressed when they have been intentionally added, such as the case of the reagents or the catalyst used in synthesis. In the case of prazosin hydrochloride monograph, the test for nickel has been deleted. It is now covered by the ICH Q3D guideline. There is also a general statement given in the general monograph substances for pharmaceutical use to put a framework for this. The test for elemental impurities will remain when those elements are of natural origin and cannot be easily eliminated by purification. This is the case of the mined excipients. And there are also some specific cases like methylene blue, where the elements may have an effect on the therapeutic activity. And in this case, you will also find tests and limits for the elemental impurities. Another group of impurities that have to be controlled are the residual solvents or volatile impurities, which are in line with ICH Q3C. They are covered by the general chapter 5.4 for residual solvents. An individual monograph does not include a test for residual solvent, except for class 1 solvent, which are the most toxic, and class 3 solvents exceeding the limit of 0.5%. For class 2 solvents, the limit is set by option 2 described in the general chapter 5.4. In the monograph of ethambutyl hydrochloride, you will find a test for the control of class 1 solvent 1,2-dichloroethane at maximum 5 ppm. And in the olmesartan medoxomium monograph, there is a test to control acetone at maximum 0.6%. A class of impurity that might be of interest for you is the DNA reactive or mutagenic impurities. Please remember that the European Pharmacopoeia follows ICH M7 guideline for active substances. And this is reflected with a general statement in the General Monograph 2034. An individual monograph might contain sometimes a production section or a test section for the control of mutagenic impurities, and this only if the genotoxicity is proven. And this can be done either by availability of AIMS tests, toxicological studies, or any safety assessment reports. Please remember that the structural alerts alone, those can be found by DEREC or other softwares, are not sufficient to prove the genotoxicity. If you would like to know and to have more information about the different type of impurities and the control that is put in place in the monographs, please subscribe to Module 3, Impurity Control in the European Pharmacopoeia. I will continue now with another mandatory section, the assay. Often physico-chemical methods are described under assay, but you can also find bio or microbiological assays. If a titration is described in a monograph, you know that it's much more precise, quick, and usually cheaper than an HPLC assay. If an HPLC assay is described, then you have to use reference standards with the site content and also fulfill with the repeatability requirements described in the general chapter 246. The choice of an assay in a monograph will depend on the substance itself, but also on the experimental verification during the monograph's revision or elaboration, and of course on the dossier submitted by the manufacturers and approved by the competent authorities. Another section which is given for information and is not mandatory is the storage section. In some cases, competent authorities may require some particular storage conditions and may decide to make them mandatory. In any case, The substance has to be stored in an appropriate way to ensure compliance with the monographs throughout the whole shelf life of the substance. Please note that you can find some conventional expressions in the general notices. I would like now to say a few words about another informative section, functionality-related characteristics. This section is mainly described in monographs on excipients. It is not mandatory, and there is a general chapter, 5.15, with more detailed information about this section. This section contains characteristics that are shown to impact the functionality of an excipient for a certain use. A monograph of excipient might contain either the characteristics or the parameters, or propose tests to control those parameters. And all the tests are linked to a certain use, which is in line with ICHQ 8. Let us see some examples together. In the case of sorbitol, the particle size distribution and the powder flow have been found relevant when this excipient is used as filler and binder in tablets. In calcium hydrogen phosphate, 3 parameters are found relevant when this excipient is used as filler in tablets and capsules. But please remember that other characteristics may be relevant if the same excipient has another use. The labeling section is generally covered by national regulations and international agreements. It is not always present in individual monographs. And usually it contains informational items or it may include some recommendations. However, for the purpose of the pharmacopoeia, there are statements that are necessary to demonstrate compliance or non-compliance with a monograph, and those statements are mandatory. This is the case of the nominal values which are given in monographs of excipients. This is my last slide, and I would like to give you some key home messages. You all know that the European pharmacopoeia monographs are legally binding. Please remember the complementarity of individual and general monographs or general chapters. Do not forget that not all sections described in a monograph are mandatory. There are also informative sections. And last but not least, the European pharmacopoeia monographs are not cast in stones. There are mechanisms that are put in place in order to revise the monographs and to keep them updated with the latest regulatory requirements, but also with the latest technological and scientific advances. Any interested party can propose a new monograph elaboration or revisions, either by national authorities or directly contacting the Secretariat through our help desk tool. I would like to thank you for your attention, and I give the floor now to my colleague Ameller for the second part of this webinar.
Thank you, Silvina, and good morning to everyone. So my part of presentation will focus on individual monographs on medicinal products containing chemically defined APIs. As you already heard, my name is Amela Saracevic, and I also work as a scientific program manager in the European Pharmacopoeia department. So let's take a look how it all started when it comes to the medicinal product monographs. In 2012, European Pharmacopoeia Commission has reconsidered its strategy about medicinal products and started with the pilot phase by elaborating several medicinal product monographs in parallel. As the outcome of this pilot phase was successful, in 2014, the strategy to widen the scope of the European Pharmacopoeia uh, was decided. So from that point on, we include medicinal product we elaborate also medicinal product monographs. And we consider this still quite recent because if you consider the 60 years of the European Pharmacopoeia and elaboration of the active substance and excipient monographs. And the first focus was on the single source products, so-called P4 monographs. And the first P4 monograph was sitagliptin phosphate tablets, published in Pharma Europa 26.34 comments. And it was adopted in 2015 and published in the European Pharmacopoeia. So 2015 and 16 could be considered as milestones because it was the publication of the adoption and publication of the first medicinal product monographs that came into force on 1st of April 2016. And another milestone that I would like to mention is 2019, when we have adopted the first P1 monograph on rosuvastatin calcium tablets. I would like to clarify these terms that I'm using. So P4 stands for the products that are still under patent, so where we only have one product on the market, those of the innovator, whereas P4 monographs are those who are elaborated based on several manufacturers' collaborations, because we have several marketing authorization on market, and generics are already present on the market. So now where we are now, as said, we started with single source product elaboration, but now we also elaborate multi-source products, so P1. We also started with the immediate release tablets and capsules, but over the years, the scope also includes now the solution for injections and infusions, oral solutions, powder for injections, and most recently, also modified release oral forms, and also combination medicinal products, so products with more than one active substance. On this slide, I have listed all the adopted and published monographs so far. So we have 26 monographs published in the European Pharmacopoeia so far. And mainly, as you see in white, they are based on the single source product, so elaborated under P4 procedure. And until now, we have published also 2 monographs that are based on multi-source products. So the work is slowly starts to increase there as well, as you will see on the next slide, in which I have listed the monographs currently under elaboration. We have 35 monographs, and in white are those that are elaborated under P4 procedure, whereas the yellow represents monographs being elaborated is P1 monographs. As I mentioned before, we also have included now the first combination single source product on our work program. This is metformin, hydrochloride, and dapagliflozin propylene glycol tablets. But also we have widened the scope to include modified release products. So the first modified release product that we are working on is esomeprazole gastro-resistant tablets. When it comes to the general policy and approaches, they are all captured in the technical guide for the elaboration of monographs on a medicinal product containing chemically defined active substances. And we are already in third edition, published in 2023. Policies have been evolved in the past years, and they continue to evolve to best tackle the needs and reflect the regulatory requirements and scientific progress. And this latest edition of the technical guide included some updates. For example, as we have added the first combination products to our work program, we have included that it's possible to elaborate monographs on medicinal products which are combination medicinal products. The policy and repeatability criterion in essay that you will see a little bit further in my presentation was also introduced as a general rule. And what we found also very important is indication of the strength of the medicinal products that was considered during the elaboration of the monograph. And this is provided to users as the information in the knowledge database once monograph is published. It's worth mentioning that for medicinal product monographs, the technical guide for the elaboration of monographs in general applies. And this technical guide that I have mentioned here, it covers only specific points relevant really to medicinal product monographs elaboration. So the important aspects were defined in the past years, as said, to define our policies. And the most recent was the adoption of the revised general monograph on pharmaceutical preparation 2619 to clarify widening the limits for total of impurities in exceptional cases. This was adopted by the European Pharmacopoeia Commission in June last year, and it's published in Supplement 11.5. So in exceptional circumstances and if justified by the applicant to the satisfaction of the competent authority, the latter may approve a wider limit than those described in the monograph. In this case, the competent authority shall bring this to the attention of the Commission to revise the monograph if needed. This slide contains several policies that were defined over the years, but I will mention them later during my presentation in more details. Important to note is also that we are engaged in the prospective harmonization with the other pharmacopoeias. This is out of scope of the official harmonization that is happening in the PDG, so Pharmacopoeial Discussion Group. This prospective and unofficial harmonization was initiated with the US Pharmacopoeia in 2008 on new active substances, so those elaborated on the P4 procedure, and this was then enhanced also to a medicinal product monographs, also for products still under patent. When it comes to numbers, so far we have harmonized 10 monographs with the USP, and 11 projects are still ongoing. And following this successful example of the prospective harmonization with the USP, this project expanded also to bilateral harmonization with the World Health Organization and the Japanese Pharmacopoeia, and now includes also P1 procedure, so the products for which generics are already on the market, so multi-source active substances and medicinal products. This started in 2023. There are many advantages of prospective harmonization, especially for the manufacturers, because it can be cost and time saving for them. First of all, they will have same dossiers, so same documents and samples that will be required by both pharmacopoeia. So it's a coordinated work, and there are always regular updates. So it's a full transparency between us, involved pharmacopoeias, and the manufacturer, and there's a joint communication. This is a coordinated work between pharmacopoeia, so we can also share our experience and knowledge. And the final goal is to have the aligned test procedure and limits to a common quality standard. At the end, final respective texts do not have to be identical, but we really strive to have the text harmonized as much as possible. So to go more in details about the general principle for medicinal product monographs, they are elaborated for medicinal products that have been authorized in at least one of the member states of the European Pharmacopoeia Convention. So the monograph specification are based on medicinal products that are currently approved. These monographs provide shelf life specifications. As for any other monograph, the tests are mandatory unless otherwise specified. the flexibility is offered by the European Pharmacopoeia and it's covered by general notices. And this is especially important because the choice of analytical procedure may be affected by the formulation and or manufacturing process. So when using medicinal products monograph, each marketing authorization holder must demonstrate that the tests described in the monograph are appropriate for the quality control of their products. As already mentioned yesterday and also by Silvina, individual medicinal product monographs, as any other individual monographs, cannot be seen as a standalone text. It's always complementary to other text, so general monographs that applies. In this case, it's pharmaceutical preparation, general notices that apply to all the text of the European Pharmacopoeia, or general chapters, because these are information on the procedures like liquid chromatography that are described, and also dosage form monographs. Monograph on the medicinal products is drafted with the same overall structure as a monograph on chemically defined active substances, and it covers different formulation and strengths of the same dosage form. It was decided that one monograph will be elaborated per active substance. So, for example, for the products containing a salt form and the base acid form were considered as different active substances. So it will be different monographs elaborated as well. Similar approach is taken for the medicinal products containing different active substance solvates. So in this case, separate monographs would be drafted. And when it comes to the hydrates, this one monograph will be elaborated for medicinal products containing one or several hydration forms of the active moiety. So these are all policies that I have mentioned previously and that were developed during the years building our experience on medicinal product monographs. So if we go into each section separately, let's start with the titles. So title of the monograph on medicinal products combine the appropriate active substance name and the relevant dosage form. Active substance name, the most commonly is the international non-proprietary name that is used. As said by the policy defined, salt is always mentioned in the title, as well as the solvates, if not hydrates. But the hydrate form is omitted. The reason is for that, it's what I mentioned previously, that one medicinal product monograph can cover the active substance of several hydrates. And for the dosage form, we use titles or subtitles of the relevant dosage form monographs. And there are a few examples like dronedarone hydrochloride tablets, lacosamide infusion, cabazitaxel acetone concentrate for infusion, and so on. The next section is the definition. And the definition specified the exact pharmaceutical form, so dosage form that is covered by the medicinal product monograph. Specific, so the active substance covered, specific salt, hydrate, and or solvate, and it gives the reference to the this relevant active substance monograph. It's also indicated that the medicinal product monographs are for human use only, and if appropriate, it can state that the preparation is sterile. What is given here, it's also the cross-reference to the relevant dosage form monograph. This is not usual to give the cross-reference to the general monographs because of the complementarity. It's to be understood, but for the clarity, and it was decided that in the medicinal product monograph it would be important to give this information. And the definition also contains the content, which is given as a percentage of active moiety declared on the label, and you can see a few examples on the slides. Production section. it's included in the monographs on medicinal products containing active substance in solvate form if the solvate is other than hydrates. So in that case, it was decided that no test for the control of that organic solvent will be described, but the statement in the production section as follows is published. So manufacturers are expected to evaluate whether the presence of the active substance as a solvate is critical to the quality, efficacy, and/or safety of the medicinal product, and where applicable, to implement a control strategy for a corresponding solvent to the satisfaction of the competent authority. If you go to the next section, which is identification, this section describes tests to confirm the identity of the active substance in the medicinal product. So the test described must make it possible to differentiate the active substance from other compounds that are closely related and likely to be present. Usually, we use the combination of the LC method and UV spectrum. So in the LC method, we compare retention time and size of the main peak. But as this is not sufficient to confirm the identity, it's always combined with the UV spectrum. The other options are also possible. Sometimes we have a combination of the liquid chromatography and infrared, or the alternatives can be given, as you can see on the examples, where the users are, for example, instructed to carry out tests A/B or test B/C, which would be a combination of the LC and UV spectrum, or LC with the infrared spectrophotometry. The test section of the medicinal product monographs typically includes related substances test and dissolution or disintegration test for tablets, capsules, and so on. If not product specific, additional tests to control specific quality parameters can also be included. For example, pH for liquid or semi-solid dosage forms when it's indication of their stability. Other tests such as bacterial endotoxin, microbial testing, uniformity of dosage units are not included because they are referenced and covered by general text, general monographs, and dosage form monographs. They are included only in individual monographs if a specific procedure, sample preparation, or a specific individual limit is required and has to be described. If we go into details in the impurity policy that we apply for the medicinal product monographs, this is in line with the ICH Q3B and Q6A. So degradation products are always controlled. So those are the impurities arising during the manufacturing process and throughout the shelf life, including impurities of synthesis that are also degradants. Acceptance criteria that are set can be individual acceptance criteria for specified impurities, or all unspecified impurities could be covered by a general acceptance criterion. So impurities of synthesis are not controlled in the medicinal product monographs unless they are degradants, as mentioned. If they are detected by the method given in the individual monograph, they are included in the transparency list. And if they are present at a level greater than the reporting threshold, they have to be identified using a CRS or a reagent, and they have to be disregarded. This is well presented in the following examples. I have taken deferiprone tablets to show you how impurities are identified and limited. So here you will see that we have several solutions described that are used for the related substances test itself, but we also have some solutions described for the assay, which means that in the monograph we'll have one liquid chromatography procedure both for related substances and assay. We also give a solution for the system suitability. In this case, it contains a reagent maltol, which is impurity B for this substance. So impurities have to be identified. So we would use this reference solution B that is prepared for the system suitability test to identify the peak due to impurity B. And it is used as set for the system suitability. So we have a resolution criterion. And because impurity B is a impurity of synthesis of the active substance of deferriprone, and it is limited to 0.10% in the API monograph. If it's present above reporting threshold, which in this monograph is 0.05%, it means that will be detected on the chromatogram. But as we said, that medicinal product monographs do not cover impurities of synthesis, this peak needs to be regarded, and this is why we provide the way to identify that, identify the peak due to impurity B to be regarded and not to be taken in the calculation of the total impurities. When it comes to the dissolution, this policy was adopted in November 2020. It was one of the first policies that was adopted for the medicinal product monographs, and it's described in the general notices in supplement 10.6. This is the exact text that you will find in the general notices, and I will try to highlight just the important aspects. So a suitable product-specific dissolution test has to be proposed by the applicant for routine quality control and to confirm batch-to-batch consistency of their product. Where appropriate, the solution test is included in individual monographs. The important part is that the applicant may either select the monograph dissolution test or develop in-house dissolution test. In all cases, the applicant has to demonstrate the suitability of the selected test, either it's the one described in the monograph or their in-house test, to the satisfaction of the competent authority. If in-house dissolution test is proposed, justification for not selecting the monograph dissolution test is not requested. However, when tested, the medicinal product has to comply with the test also described in the individual monograph, unless otherwise justified by the applicant. Quantitation in the dissolution is usually done by the liquid chromatography or UV, using either reference standard with assigned content, for example, in rosuvastatin calcium tablets, or specific absorbance value if we use the UV for dronedarone tablets, for example. Disintegration test may be substituted for a dissolution test in accordance with the ICH Q6A, and this is outlined in the general notices as well. So for rapidly dissolving medicinal products containing highly soluble active substances throughout the whole physiological range, and when there is a relationship to dissolution is established, or even when disintegration is proven to be more discriminating. So the first monograph published on sitagliptin phosphate tablets actually contains disintegration test. Such a substitution, of course, has to be justified by the applicant to the satisfaction of the competent authorities. The part on the assay is so medicinal product monographs must include specific and stability indicating assay to determine the content. Usually this is the LC procedure, and the same procedure, whenever possible, the same procedure as that described for the related substances is given. And the standard specification includes 95.0 to 105.0% of the content stated on the label. So this was one of the policy that was also recently added to our technical guide, that repeatability requirements of the chapter 246 are only valid for the active substances. It was decided to include the criterion for repeatability in each medicinal product monograph, and the policy approved states that the limit relative standard deviation of 1% determined on six injection will be included. So this is the general rule and can be adapted depending on the values reported, so during the verification of the monograph's results obtained. When the reference standard of the active substance monograph is used, conversion factor may be required. For example, rosuvastatin calcium, CRS is used for the determination of rosuvastatin in rosuvastatin tablets. In that case, because we have specification that indicates the percentage of the active moiety, we need a conversion factor as well. We also have a transparency list of the impurities, the same as in the active substance monograph. And we list all impurities independent of their nature, so both degradants and synthetic, if they are known to be detected by the analytical procedure given in the monograph. Impurities that are relevant to the active substance, so the common, and they keep their designation, so impurity A, impurity B, and so on. But if we have impurities that are specific to the medicinal product only, they will be designated by FP followed by the letter. So it will be FPA, FPB, and so on. So at the end, to conclude, as you could see, a number of monographs have already been elaborated under the P4 procedure, and there's increasing number of the P1 procedure being used for the elaboration on medicinal product monographs as well. We are engaged in the prospective harmonization with the USP, Japanese Pharmacopoeia, and WHO, and this is ongoing for various projects with the aim to reach the aligned standards. And policies for different parts of the monograph, such as titles, definition, identification, assay, impurities, dissolution, are now well defined over the years, and this is all captured in the technical guide. Indication of the strength of the medicinal product that is considered during elaboration is provided to users for information, and this is one of the recent policies that was decided to help the users. And as for any other monographs, the medicinal product monographs are also continuously revised to keep pace with the regulatory requirements and scientific advances. So you can always come back to us to propose either a new monograph for the elaboration or to revise already published monograph. I think that I reached the end of my presentation, and I thank you for your attention.
So if you agree, we can take the Q&A session now. I can start already with some questions that we have received. First question, is it possible to have specification for liquid chromatography tighter than 98.0 to 102.0% if development studies has demonstrated that variability of the method is lower than 2.0%? In theory, it is possible. It will really depend on the approved specification. As the limits in the monographs are based taking into account the approved specifications, experts could not go beyond those approved specifications. Those are the safe specifications. So if different limits are approved by competent authorities and we get a dossier with those limits, of course, we will take them into account and reflect them in the monograph. Another question, if the monograph specifies only anhydrous basis, can we calculate assay on solvent-free basis? Yes, you have to. The individual monograph only give information about the anhydrous basis or dried basis, but you always have to take into account the residual solvents and take this for the calculation. Again, this concept is covered by a general monograph and not the individual, but you have to take into account the water content and the solvents for the calculation of the content. Another question received, the characteristic section or the character section on solubility, etc., does not need to be performed. I would say it is better recommended to perform this section. This will already give you an indication if there is any issue with your substance. If the solubility is different than those mentioned in the monograph, it means that probably there might be any issue at the same time, again, it is only informative, so not analytical requirement. I know that in some cases, competent authorities might require the compliance with this section. So if the competent authorities require this, then the marketing authorization holder has to comply with all the requests from competent authorities. However, again, the section on characters is only informative. Can CP, so I guess a certificate of European Pharmacopoeia monograph, be replaced of GNP by an national NRA? I'm not sure I can understand correctly this question. So you have to put in place some GMP system, and the CP is not the same as could not replace the GMP. There is a specific module focusing on certificate on CPs, certificate of suitability with the European Pharmacopoeia. So I can encourage you to follow this module for more details. Another interesting question. You mentioned that information on production is mandatory in monographs, but not every monograph contains this section. How does this fit together? This is completely right. It is a mandatory section if it is present in the monograph. And again, this section would be included in a monograph depending on the information received by the dossier submitted by the companies and approved by the authorities. So if there is a really particular attention to be paid, like in case of pethidine, where there is specific requirements if this substance is used for parenteral use, then we will include this section. It really depends on the substance and on the requirements, and of course this will depend on the dossier which are approved. I hope I have responded to the user. So another question regarding identification tests this time. Are the identification tests mandatory to be performed? I'm talking even for first identification. Yes, identification is mandatory section. First identification and all the tests described under the first identification are mandatory. They are intended for pharmaceutical industry, and they can be used in all circumstances. Next question, what is ODI stand for in unspecified impurities? So ODI in one of my slides means other detectable impurities. So the unspecified impurities are also code, and you can find these terms in some general monographs, other detectable impurities. Another question with respect to identification. Could I make second identification instead of first in pharmaceutical industry if there is a shortage or cannot do all first tests? This may only be done if competent authorities authorize you to do so. So first identification is the one to be used and intended for the pharmaceutical industry. If for some reason there are issues to perform this identification, you have to contact competent authorities in Europe and have obtain their authorization to deviate from the monograph and perform, in this case, second identification. We cannot give you such a authorization or permission for deviation. Only competent authorities are those who may give you a permission for deviation or may impose additional requirements if considered necessary to ensure the quality of a substance. Another question, can we keep an in-house impurity without identifying it with a limit of 0.15% as RRT impurity. So if you have any in-house impurity which is not listed in the European Pharmacopoeia monographs, it have to specifically at a limit above identification threshold, again 0.10 in case of a maximum daily dose below 2 gram for the substance, you have to propose the suitable analytical procedure and limits and control this impurity and submit all the data to competent authorities for assessment. If authorities evaluate these additional impurities and they approve your requests to control those, they will also contact the pharmacopoeia in order to let us know about this additional impurity or impurities and asks to revise the monograph in order to update it and take into account new sources on the market. I have a specific question on gelatin monograph. I will ask the users to contact and submit the question through help desk because it is really quite specific for a certain general method 2223. So if you contact us through help desk, the question will be submitted to the scientific officer in charge of the monograph's elaboration, who will be able to give you really more concrete and relevant information to your question.
Would you like me to take some?
Yeah, Amela,
yes,
if you would like,
I think,
go ahead.
So how do you choose which medicinal products need a monograph elaboration? Well, to come to the work program of the concerned group of experts for the medicinal product monograph, it's exactly the same as for the active substance monograph or excipient monograph. So anyone can make a request. So manufacturers and other interested parties, those coming from the member states of the European Pharmacopoeia Convention, have to do that via their national pharmacopoeia authorities, and for the others, so manufacturers and interested parties that are outside of the member states of the European Pharmacopoeia Convention can write directly to us, so to the EDQM Secretariat, EPD Secretariat, via help desk. So anyone can propose a monograph for the elaboration, including medicinal products monograph, but it's also very important to know that we need to have collaboration of the manufacturers, because monographs and as always, the prerequisite, so basis, is that this medicinal product is authorized in at least one member state. And again, to reiterate that we need really collaboration from manufacturers, because we would need dossiers, complete dossiers of one or more manufacturers, depending if it's a single source or multi-source product, and we would need samples, because before publication, monographs of the European Pharmacopoeia are always verified in at least 2 laboratories. So we need also samples of impurity, active substance, medicinal products, and so on. Is it possible to start a prospective harmonization also for API monographs? I think that I have mentioned that with actually it has already started. So with the USP back, I think in 2008, it already started with the single source active substances. So those elaborated under P4 procedure, and then continued with the medicinal product monographs as well, being harmonized with the relevant pharmacopoeia. And last year we have started projects of bilateral harmonization with the Japanese Pharmacopoeia and WHO, also both on the APIs and corresponding medicinal product monograph. Just to scroll a little bit to see something relevant for the medicinal product. So how is it possible? What limits of total impurities, if medicinal product monograph, including it. So the limits that are given in medicinal product monographs are those limits that are approved on the European market. So the total impurities also come from the approved specifications. If a product monograph provides a disintegration test, is applicant allowed not to collect the solution data? I tried to explain. So if you replace the solution by disintegration, this is possible with for these substances that are rapidly dissolving and to contain highly soluble active substances. But this replacement, so substitution, has to be justified by the applicant to the satisfaction of the competent authority. So only if it's authorized by the competent authorities, you can have this integration test to control your specific product. This as API manufacturer to be included in the EP monograph, the product must be marketed in Europe. Are there any exceptions? I think this is really the basis of all our elaboration, that the active substances has to be in the authorized product on the European market or in the member states of European Pharmacopoeia. I think that I also saw some other questions. I'm scrolling down because it's a mixture of the questions for Silvina and could you confirm that combination product only means product with more than one API, not also API plus medical device? And as you expect, the answer is yes. So when we speak about the combined product, it means product containing more than one active substances. So far, there were no discussion on the example that you gave with the combination of the API and medical devices. So this was not part of the discussion of the European Pharmacopoeia Commission so far. Okay, I think that I have seen some, yes. When European Pharmacopoeia and USP methods are harmonized, is it acceptable to use only the USP reference standards for the test? What I mentioned in my part of the presentation was a prospective unofficial harmonization. So in that case, the methods that are given are not interchangeable. So the aim of prospective harmonization is really to help the manufacturers for the time saving, cost saving, to exchange our knowledge and expertise with the other colleagues and really to come to the aligned standard. But this is not official harmonization. When it comes to the officially harmonized standards, so those that are under the PDG, they are only interchangeable. The prospective harmonization that I spoke about is really unofficial. So at the end, if you want to demonstrate the compliance with the European Pharmacopoeia, you have to use the European Pharmacopoeia tests and specifications. I don't know, Silvina, I think that you have more. If you want to continue,
Yeah,
I will search
I
if there
can
are other for
continue,
me.
of course.
Yes.
Thank you. So interesting comment, question or comment. I would like to share microbiological testing is not included in the monographs. You also have to have the following general monographs requiring microbiological testing 2034 substantive pharmaceutical use at general chapter 5.1.4 for non-sterile pharmaceutical preparations and monograph 2619 pharmaceutical preparations. Yes, what is important again to remember is this complementarity of the individual and monograph and general monographs and chapters. So you have to comply with the requirements of the individual monographs, but there are also others relevant general monographs and chapters, and some of the requirements are put in those general monographs and chapters. So that's why it is important to, when you start with the compliance of an individual monograph, also to know which are the relevant general monographs and chapters where you can find the additional requirements which have to be applied. The case of the microbiological testings, which is covered by the different general chapters and monographs. Okay, another question. In some cases, to reach the SST acceptance criteria is not easy due to technical limits. In these cases, there is a direct way for having more detailed technical suggestions than those reported in the monographs. If there is any specific recommendation to be given to our stakeholders and found during the experimental verifications by the experts, those would be given on the knowledge database. Knowledge database is not legally binding, but it can contain really useful information. Sometimes you have information of procedures about rinsing or any other technical issues that have been encountered during the laboratory verification. On the other hand, if there is nothing given, then it means that the tests have been verified with no issues. Specifically for the SST, if you have any difficulties to comply with, you can also adjust conditions, and this adjustment of conditions, which is permitted to some extent, is also described in the general chapter 246. Of course, if you still can't reach the SST, you can always contact us through our help desk too to search for more support. I can take who can apply on elaboration on monograph of an API, marketing authorization holder or API manufacturer. So for the elaboration of monograph on active substance on API, we need the collaboration with the active substance or API manufacturers. They will collaborate with us and supply us with the data and the samples for the verification of the method.
So I have again a similar question for the complementarity. Okay. So for final products, the marketing authorization holder should apply the prescription of both monographs, medicinal product monograph and general monograph, and this is correct. So we spoke both about complementarity of individual monograph that always has to be seen together with the all relevant general monographs that applied. So for medicinal product, this is mainly pharmaceutical preparation and the relevant dosage for monographs. So yes, you have to comply with both requirements that are given in the individual monograph and those covered by the dosage forms general monograph. Mm-hmm. For P4 monographs, will these be revised once products from several sources will be available? Well, this is usually the case when the P4 monographs, so the monograph that is elaborated under this procedure only with when single source product was present on the market. When others companies come on the market, we usually do get requests for revision, for example, to include some additional impurities, to revise some tests, and so on. So all the work starts when there is a request for revision. So it's not that immediately the monograph will be revised when the generics come to the market, but we need to have a request for revision that would be approved by the European Pharmacopoeia Commission, and then it will be allocated to the relevant group of experts dealing with this revision when there is a need to revise a monograph.
I can take some questions.
Yes, please go.
If an impurity limit is not complying with the European Pharmacopoeia limit, then what measures can be taken by the drug substance manufacturers? So if there is any issue to comply with the limit, you have to contact competent authorities and submit data and justification if you would like a different limit. And if the competent authorities, after assessment, approves this different from the pharmacopoeia limit, then they will contact us in order to trigger a revision of the monograph. Again, as I mentioned, at a certain point, the monographs are not cast in stones. When there is a need, when it is justified, approved by competent authorities, limits are updated. They can be tightened if it is necessary to ensure the quality, or they, in some cases, can be have a higher limits, again, if it is justified by the process, or if there is a new source on the market. But it is very important to contact competent authorities.
There's another, sorry.
Yeah, no, I will take two more questions. So I have suggested a relative retention in a monograph only for reference purpose. This is correct, but cannot be used for identification. But what if respective CRS is not available? Yeah, unfortunately, this is very valid question. We have old style monographs where we will not, unfortunately, propose a chemical reference substance for the identification of the specified impurities. And we try to collaborate with manufacturers to update those old style monographs. In any case, either you have to sanitize the product, the impurities, inject them to know the relative retention, or again contact the authorities to have authorization to use other ways of identification of specified impurities.
Sorry to interrupt. We have 4 more minutes.
Okay, thank you.
Yes, thank you.
Another very interesting question is about nitrosamine impurities. Any future plans to include NDSRI or nitrosamine impurities as part of the API monographs? There was a general statement include in the general monographs 2034 substances for pharmaceutical use and 2619 pharmaceutical preparations about the control of those nitrosamine impurities. And last year, the European Pharmacopoeia Commission has agreed on a policy regarding those impurities. This policy is given on our website, and as a general and active substance monograph will not include a test for nitrosamine impurities. Again, this is covered by the general monographs 2034 and 2619. So I can encourage you to go to our website to learn more in details about this new approach and strategy for control of nitrosamine impurities. And tomorrow, no, sorry, not tomorrow, but Thursday, there is the module 3, impurity control in the European Pharmacopoeia, where you can find also more information about the different the different impurities and different control put in place by the European Pharmacopoeia. So Amela.
I also have it's a more general question on the basis of what criteria are products selected for P4 monographs. I also try to explain what P4 stands for. So P4 procedure is used when we have only single source products, so active substance and medicinal product on the market, which means there is only one approved product, those from innovators. And you will also have all these slides and registered presentation after this webinar. I saw several questions on this, and this was also explained at the beginning. You will receive, and in these slides, you will find some links that you can also use to get more information. And one of the links provided is also about P4 procedure. So I invite you to look at the presentation afterwards if you need more information on the specific P4 procedure. And also, is it possible to use general monograph for medicinal products, even if there is no monograph for the medicinal products to have a good basis? Well, this is well defined in the European Pharmacopoeia when it comes to general monographs. So general monographs cover the classes of products, and if there is no restriction in the scope of these general monographs, then it is applicable to all the products in this defined class, even if there is no individual monograph. So irrespective of that, whether there is an individual monograph or not. So for the medicinal product, you have to use a dosage form monographs for control of your products. And I think, yes, that I believe that for any other questions that you have asked and we did not manage to answer because of the restriction of time, you can always use our help desk platform to send your questions to us, even those more specific. Then, as Silvina mentioned, they might be attributed to our colleagues to give you more concrete answers on. So thank you very much once again for joining this webinar today, and follow those that will be on Thursday and next week as well.
We hope that we have brought some clarification, at least to some issues, and with respect to the individual monographs, and again its complementarity with the general monographs and chapters. Thank you very much.
Thank you. Bye bye.